Greeting to everybody. This is the first part of the applied genetic in the laboratory animal science. The title is Genetic Quality Assurance, or it could have been also genetic standardization. You can see in the study plan, which is bold, this is the part 1. And the normal typing is the material of the second part of this lecture in a separate YouTube presentation. So you can see in the that after an introduction we will see the two lines of the genetic standardization, namely the genetic uniformity, which is part of this first lecture, and the genetic variation, which is part of the second lecture. The second lecture will be supplemented with the genetic quality control, the calculation and the representation of the genetic relationship between inbred strains, and the question of biobanking. Before really starting, please let's examine that people of the audience in this room or in the imaginary space are appropriate for a possible experiment by dividing in two groups, a control and a test group, and receiving a drug medicine candidate or so. No, because all of us is in conventional microbiological state. Everybody is different in this respect. Our genetic background is mostly unknown, but what is sure that this is also very variable. We have different home and different eating habits, eating diets, with a lot of influences and sources of standard deviation. What is common that all of us are apes. So the conclusions that if we are participating in a trial, similarly to the animals, we are a black box and we receive some influences and we give some biological answer. But because the material is very, very variable, the number of participants should be extremely high, so this is not a real experiment. This would be the question in case of the animals, but we can help by standardizing the different environmental factors like the feeding, the keeping, etc. But first of all, the genetics. So the deviations of the data from animal experiments mostly derives from the genetics, from the genetic differences. Therefore, the genetic background of the animals, of the experimental animals, should be standardized, which can be made by a special breeding system or by very, very modern and sophisticated biotechnological methods. We can go in two directions, forming animal apt to experiment. First, to make animals more and more uniform, which help us to receive more and more uniform biological answer. But in this case, the whole population will not be covered. The second possibility is to form a variable population which most more reflecting the human population. So what are the first line, the first direction? The direction of genetic uniformity. First of all, because this is historically important, but not the first line in the today modern laboratory work, is the monozygotic animals and the twins. You can see twins in the picture, calves. They are monozygotic twins. And as a extra double twins can be found in the nine-banded armadillo, where we have genetically identical 4 offsprings per litter. So the siblings of the nine-banded armadillo are genetically identical. The armadillo produces 4 monozygotic offspring per litter, derived from 1 fertilized egg. By the way, what is the nine-banded armadillo for in the experimental work? So in the experimental work, they were used for leprosy research. But before given the details, I have to finish this slide. So what you have to check on it, you have to see that the twins calves are not absolutely identical in the repartition of color. And the third possibility can be found also in this slide, the cloning of the animals. If all the individual will be the clone of the other, which is a very useful and modern process, and it is in use in case of cephalopods when all the experimental animal in a group can be a clone of each other. But returning to the leprosy and the nine-banded armadillo as a history in laboratory animal science, and hopefully more and more only history for the humanity, but not totally. So the leprosy, or the synonym is the Hansen disease, is a slow forwarding tuberculosis. This is a disease caused by Mycobacterium leprae, which is a slow moving bacteria, and the nine-banded armadillo is an excellent model animal for this disease. You can see that both the skin symptoms and the neurological and heart symptoms can be found, can be form a model. But returning to our original questions, that the calves on the picture are totally identical, so the twins are totally identical. No, they have phenotypical shown differences in the coat patterns, because in the intrauterine life they receive different influences. Subtle differences in the environment during the intrauterine development may have caused variation in the migration of melanocytes from the neural tube to the peripheral part of the body. And the conclusion is difference in the coat patterns. This is the same in the brain gyruses. And in genotypical plane, we may have also differences, namely in the epigenetic signs, in the DNA methylation pattern. The mostly used form to increase the uniformity of experimental animal is the use of inbred strains. By definition, we can produce inbred strains by crossing of closely related animals. It can be a brother-sister mating, or, if necessary, the mating between offspring and the youngest parent. It was discovered by Carl Jensen that after a lung tumor graft, there was no rejection if the population was inbred mice. It showed that with these techniques we can make a uniform population. The inbred strains mean that the coefficient of the inbreeding is very high, close to 100%. This coefficient of inbreeding, or F value, is the fraction of the original heterozygous genes which got fixed in homogeneous state during the brother-sister mating. Here you can see a graph about it. You can see 1 up to 20 generations, and the increase of the F value, the increase of inbreeding per generation, which depends upon the relationship between the individual pet partners of the breeding pairs. In other words, it's depend upon the consanguinity. In the A line, where there was a brother-sister mating, it results in a steep increase of the inbreeding coefficient from generation to generation. In the B line and C line, they used random matings, and the number of breeding pairs in case of B used to be 5 and C 8. And you see that in this case, in random meetings, the increase of the inbreeding coefficient, the F, depend on the size of the population, and if the population size is high, you cannot move on towards the uniformity. So this is the basics, and here you see the symbols of the chromosomes of inbred animals. You can see that there can be two types of homozygous animals. Number 1 and 2 left and in the middle are isogynous to each other, because not only the allele pairs are the same, but they are identical, but in the middle and the right side they are anisogynous, because the two chromosomes or the two animals is not the same, but they are homozygous animals. The increase of the inbreeding coefficient, the delta F, can be expressed in equation. When the matings occur at random, the delta F per generation will be 1 per 8 females plus 1 per 8 males. Uh, and if the number of males and females is the same, the delta F is 1 per 2n, where n is the population size. In the inbred strains, between the 8 and 19 generations of the brother-sister mating can be splitting, and parallel lines are found. Or if differences are detected between these parallel lines, or if a branch is separated and maintained at a different laboratory or site. So in the background is a genetic drift, and the genetic material will a little bit different. So the cause, the genetic drift, means that undetected fraction of alleles may become progressively fixed in a homogeneous state, replacing the original alleles. This process is known as genetic drift. How to maintain the inbred strains? The inbred strain can be maintained in form of unline breeding, when from the founder parent pair and from each further parent pair only a single brother-sister pair is selected for breeding. This is very strict, but in the practice they do that inbred strains derive from some founder parent pairs. They use in the maintenance traffic light system. The crossings, the matings in the practice, if not between litter mates, it should be at least within the generation, and the cages are marked by small color plates, and in the convention the white is for the founder, the green for the filial generation, the yellow for the second, and the brown for the third filial generation. So what can be kept, that the crossings should be within the generation. What is the biological effect of the inbreeding? The biological effect of the inbreeding is first that animals carrying of detrimental recessive alleles will be eliminated. The respective line will decrease in biological vigor. So we can see that till 10, 13 generations, the less favorable combinations of alleles will be eliminated. The remaining will show a decreased vigor, growth, survival, and fertility, which is called inbreeding depression. And what is also a consequence, that less genetic information than in case of the whole species, the human as a genome can be represented by 16, 17 inbred mouse strain. McLaren and Michi in 1956 exposed experimental mice to anesthetic, namely to pentobarbital. The offspring from mice of a cross between two inbred strains responded more uniformly than the parents, unexpectedly. So the crossing showed a more uniform biological answer than the parenteral inbred animals. The nomenclature of the inbred strain is name/breeder or holder. The name consists of 1 up to 4 capital letters. For example, A, D, B, A, W, A, G. The name of substrains should be given after a slash after the name and the substrain designation. For example, C57BL /6 and G. We may have some exceptions because they are old conventional names like C57BL, so which is not only capital letters, or C3H or 1 to 9. Symbols or synonym is abbreviation. Can be given. So I give some symbols of some strains when the A key R can be given as symbol of A key, the bulb C as C, C57BL6 as a B6, and the DBA diluted brown agouti, this is the abbreviation, 1 is D1. The other line of the uniformity is the population of F1 hybrid. Animals resulting from a cross between two inbred strains are genetically uniform. They are heterozygous for all the genes for which the two parental strains differ. See the hybrid mice. The production of hybrid seed involves 4 separate steps. There was the model for animals, the plant. So the production of hybrid seed involves 4 separate steps: development of inbred lines and testing of these in hybrid combinations. Second, multiplication of seed of the component lines. 3. Seed production of the foundation single crosses. And 4. Production of double-crossed seed used for commercial planting. So here you can see the figure of the original hybrid mice production and the illustration of the F1 hybrid, which is uniform but heterozygous for all the alleles, as I mentioned, used to be different. So the F1 hybrid is very, very uniform but heterogeneous for all genes for which line parental strains differ. And you have to make it once more and once more because the F2 generation is different. So compared to the previous illustration, the heterozygous animals, here you see 1, 2, 3 chromosomes or animals, symbols. They have black and white heterozygous loci, but they are isogynous to each other, 1 and 2, and maybe anisogynous to each other, see second and third chromosomes. The biological features of the F1 is the so-called heterosis, or the so-called hybrid vigor, the increase in such characteristics as size, growth rate, fertility, and yield of a hybrid organism over those of both parents. It's very important. Plant and animal breeders exploit heterosis by mating two different purebred lines that have certain desirable traits. The first generation offspring generally show in greater measure the desired characteristics of both parents. This vigor may decrease, however, if the hybrids are mated together, so the parental lines must be maintained and crossed for each new crop or group desired. The larger number of the allele is in the background of this hybrid vigor, and higher adaptability or resource. They are so-called resourceful animals. Generally and mostly for morphological parameters, they are more uniform. They give a more uniform biological answer. See previously the McLaren and Michi trial with the lung cancer. And they show a less sensitivity to environmental changes. In behavioral study, a more variable response can be found, can be detected, because of the higher adaptability. The so-called Tryon effect is very important to mention from this point of view. Tryon, original trial. Tryon created an experiment that tested the proficiency of successive generations of rats in completing a maze. He initiated the experiment by exposing a genetically diverse group of rats to the maze, labeling those who made the fewest errors bright and those with the most errors dull. Tryon then mated the bright mates with bright females and the dull males with dull females. Similis gaudet. After their children mated, Tryon repeated the maze test with them and again separated the bright and the dull, again breeding bright with bright and dull with dull. Tryon continued this process for 7 generations, creating 2 distinct breeds of bright and dull rats. This is the reason he showed that in the so-called intelligence, maze intelligence, there is a very important genetic background. So under certain circumstances, the F1 hybrids show less uniform response in the maze than individuals from the parental strains, and this is a reason improved ability of F1 hybrids. So it means that they have a better vision or space memory also. Here you can see the original graph of Tryon after 7th generation. On the X line you can see the number of errors, blind LE entrance in the maze in 19 trials. And left you see the number of rats, and you see that the bright, the red rats made no more than 15 to 20 errors, opposite to the dull rats with 80 to 100 errors. So we can state that the learning performance, the peripheral vision was different. Tryon's famous rat maze experiment demonstrated that the difference between rat performances was genetic, since their environments were controlled and identical. So as a conclusion for the laboratory animal science, that for the behavior studies sometimes the inbred strain are better than the F1. Further characteristics of F1 generations: reciprocal cross is possible. F2 generation loses its isogenicity. In some cases we need these generations. This is caused by segregation and recombination of the genes, of the chromosomes and genes. Okay, the nomenclature of the F1 population is maternal strain, X capital, and paternal strain. What is the reciprocal cross? A reciprocal cross is a breeding experiment designed to test the role of parental sex on a given inheritance pattern. Reciprocal cross is a kind of crossing strategy, which means to make crosses between a pair of parents, A and B, by using them in turn as female parents and male parents to obtain two reciprocal crosses of A crossed B and B crossed A. So this is very important in genetic work of a laboratory. The next possibility is the co-isogenic strains. Mutant are found in an established inbred strain. Subline, which differs from the original inbred strain by only one gene, is called co-isogenic. For example, bulb C renew, a bulb C strain carrying the nude allele and maintained in Rijkswijk. So the symbol is, as you see, for a co-isogenic strain consisting of the full strain name and substrain designation, followed by a slash and the indication of the mutant gene. Characteristics of co-isogenic strains: the mutant gene can be fixed by multiple backcrossing. Subline will differ from the original inbred strain by only one gene. In case of mice, important are the nude, the SID, the severely combined immune deficiency, the hairless, the diabetes, the obese, and the X-linked muscular dystrophy gene and lines. In case of the rat, the gene caused the rat nude rat and the Zucker diabetic fatty. Here you see another one gene induced constitutional disease in mice. We have the sex-linked anemia, we have the mentioned diabetes and obesity, we have prolinemia and muscle dystrophy. The coisogenic strains can be maintained in coisogenic strain form, but if the mutation involves a recessive allele which causes infertility or which is lethal for the progeny, we have to maintain in heterozygous state using carrier animals. Homozygous form comes from the intercross of carrier animals. The efficacy is 25% according to the Mendel rules. If we have a population with a interesting genetic trait with a gene, we can introduce these genetic traits into an inbred strain by repeated backcrossing. The resultant strain is so-called congenic, genetically similar to the original strain, but it contains the donor gene and some concomitant passenger genes. So this will be the greatest difference compared to the coisogenic strains. The nomenclature is according to recipient strain, full stop, and donor strain name. For example, B10 full stop D2. Here you can see a figure of a system for the production of congenic strains, and in the next you can see that we have a donor and a recipient strain. In details, this intercross, backcross system for the production of congenic strains, in this picture you can see the introduction of a recessive mutant. After an initial cross, a cycle of intercrossing, backcrossing is repeated at least 10 times, whilst ensuring that the animals selected for backcross carry the gene of interest. In our case, in the picture, the donor gene MN, as a result of crossing over during meiosis and the selection of the proper backcross animals, the donor gene is introduced into the genome of the recipient strain. So this is the explanation. And the resultant strain is called congenic. The next possibility is the genetic uniformity is the genetically altered strain or transgenic strains. They are created by the introduction or by the deleting of a florin of a gene. This is an insert or knockout into the germline of an inbred animal or from the germline of inbred animals. The methods, and I concentrate to the introduction, micro-injection using virus vectors or embryonic stem cell technology. Here you can see the microinjection with the holding pipette and the injection needle containing DNA solution, and you can see the fertilized egg with the polar body nucleus, female and male pronucleus. Cloned DNA into the male pronucleus of the fertilized egg cell should be transferred. This transfer into this egg should be transferred into pseudopregnant females. After birth, screening for the integration into the genome should follow using southern blot analysis or PCR. What is the so-called recombinant DNA? Recombinant DNA or rDNA is the term used to describe the combination of two DNA strands that are constructed artificially. A cloning vector is a DNA molecule that replicates inside a living cell and is used to form recombinant DNA. The cloning vector is usually a small part of the DNA strand that holds the genetic information that is needed for the replication of cells. Polymerase chain reaction (PCR) is another method that can be used to replicate a specific DNA sequence and create recombinant DNA, which is used to replicate DNA in a laboratory test tube. So you can see left on the picture a cloning vector and a foreign DNA. In the middle, ends of vector and foreign DNA fragment anneal and are mixed with DNA ligase. And right side you see the ready to inject recombinant DNA. This is a standard method of making recombinant DNA, which involves choosing the appropriate host organism and cloning vector, preparation of vector DNA and DNA to be cloned, creation of recombinant DNA in the middle and right, and introducing of recombinant DNA to host organism. After that, screening for recombinant DNA with specific properties sought for host organism. Once the DNA integrated in the genome, it will be present in all nucleated cells, including gametes, and therefore will be inherited in a normal Mendelian fashion. The nomenclature include recipient strain hyphen letter TG and N and XX and Y. TG stand for transgene, N for non-homologous insertion, and XX insert designation. For example, C55BL / sex G TGN XXY. The whole process is bioethically disputable because so-called faulty product can be found during the production, some cases up to 95%. It means that undesirable, harmful, even painful side effects like arthritis, viability, less viability, higher sensibility to stress, or diabetes can be found. A good example is the growth hormone mouse at your left. Transgenic mice expressing hybrid genes composed of either human or bovine growth hormone coding sequences fused to the mouse metallothionein 1 promoter show high serum level of heterologous growth hormone, increased growth rates, and final adult size, but at the same time decreased life expectancy and a variety of pathological changes like visceromegalo. In the middle of the picture you can see glomerulosclerosis, hepatocarcinogenesis in many cases. So you see that left normal, right the growth hormone mouse, and there is a disturb in the development of the nervous system because the so-called writing reflex doesn't work normally. If you place the rodent on its back and measuring the time it takes for the animal to right itself, this is the writing reflex, which is deteriorated in case of this transgenic mouse. In the 63 per 2010 EU directive, the breeding of transgenic animals with painful phenotype is a so-called procedure in common language, an experiment. Therefore, you have to have an allowance to breed those animals. This is the first end of the first part of the applied genetics in laboratory animal science, and in some minutes will be continued, or you can see the same title and second part in the following.